In the second week, once implantation has started and everything is successful, you start having the bi-laminar disc formation. And it is made up of this blue part known as the epiblast and the yellow part known as the hypoblast. Okay, so that is what we call the bi-laminar disc, bi-laminar embryonic disc. Then on top of the epiblast, you find this primitive streak, and then that point there is the primitive node. Okay, you have the head region and then the tail region. This is actually what is going to develop into the baby. This primitive streak you find there is going to help in what we call gastrulation, which occurs in the third week. In the third week, we are going to have 3 germ layers, an ectoderm, a mesoderm, and then an endoderm. And this primitive streak is going to help transport some cells from the epiblast will move into this primitive streak. So again, second week, you have a bi-laminar disc made up of the epiblast and hypoblast. This is the primary yolk sac. Remember, you had a blastocyst cavity. A membrane develops to line it, and then that blastocyst cavity is converted to a primary yolk sac. Then later in life, the primary yolk sac will be converted into the secondary yolk sac. Now, the layer that lines the blastocyst cavity to convert the blastocyst cavity into the primary yolk sac is known as the Hughes membrane. That's yellow membrane you see there, the Hughes membrane. That is extraembryonic mesoderm. The mesoderm has filled the space between the cytotrophoblast and then the layer of the primary yolk sac. Okay, so that is what you have here. Then again, a space will appear within this extraembryonic mesoderm. That's the space you find there, and it will split the extraembryonic mesoderm into 2. We have the splanchnopleuric and then the somatopleuric. So that is somatopleuric, which is closer to the cytotrophoblast, and then the splanchnopleuric, which is closer to the secondary yolk sac. Okay, and then you have the extraembryonic coelom or the cavity. So again, that is epiblast, and then cells, the arrow is showing you the movement of the cells from the epiblast to form the tri-laminar disc. So you can see all the arrows you see here are showing you how the cells that are coming from the epiblast are moving. Some are moving towards the head region, some are moving to the lateral side, and then posterior. All right. So again, that is the epiblast. The arrow is telling you direction of movement of cells. That is the primitive streak I told you about. That would be the amnion, or the amniotic membrane, which actually forms a roof around the epiblast, which will later become the ectoderm. Okay, and it will be filled with fluid. So the amniotic fluid will just be on top of the epiblast. Okay, and these cells that are moving, some are going to be in the midline, and then some will also displace the hypoblast. Remember the hypoblast that formed in the second week bi-laminar disc? The cells that are coming from the epiblast, some will displace the hypoblastic cells to become endodermal cells. Those that will remain between the epiblast and these endodermal cells will become mesoderm. Okay, so at the end of the day, whatever is left in the epiblast that did not move, any cell that did not move will remain and become the ectoderm. At the end of the day, you are going to have ectoderm, mesoderm, and then endoderm. So that's what you find here: ectoderm, mesoderm, endoderm, and that's the yolk sac. Now, we will talk about neural tube formation later on, but this is the neural plate which occurs within the ectoderm. It's a thickening of the ectoderm. That is the amniotic cavity with the amniotic fluid, and that is the amniotic membrane or the amnion. All right. So the mesoderm will differentiate to form these 3 layers: paraxial mesoderm, intermediate mesoderm, and then lateral plate mesoderm. That is the endoderm, that is the ectoderm. The ectoderm thickens to form the neural plate, and then eventually it can form a tube known as the neural tube. This is what will form the brain and spinal cord. So we'll touch on that later. This is the notochord. Okay, the cells that move from the epiblast, those that fall in the midline. Okay, one of them is what is going to form the notochord. All right, so also showing how movement of cells are going on. So thank you very much, and I hope this video helps.